I'm glad to see everybody else is. Are you sharing? I don't see it sharing. Yeah, is it being shared? Okay. Okay. I'm glad to see you all as keen to have a beer as I am. Everybody's sitting and ready. I don't even have to chase you. It's wonderful. I have two quick announcements. The first one is I'm really sorry about the drilling. I can't do anything about it. It is what it is. That's the first thing. And the second thing is there is quite a lot of T-shirts left. Everybody is due to get a gray T-shirt. So if you haven't received a T-shirt or picked up your T-shirt, please go so poor Lisa doesn't have to stand there the whole time of the conference. So go and get your T-shirt. It's a part of the registration fee. You don't have to pay for it. Some of you actually asked for extra T-shirts. You can also pick those up. And without further ado, I think we're on to the next session. One of the next part of this session. And I think Jacob is going to take over because somebody has to keep Kieran to time.
Yeah. All right. Welcome back, everyone. I'll be taking over from Kieran this session. Somehow two of his own talks made it into his own session. Don't know how that happened. Carrying on the same novel ideas with Ecopath with Ecosim. Up next, we have Carolina talking about exploring invasion processes and ecosystem effects, simulating marine invasions using recipient ecosystem traits and ecological interactions. I just remind everyone, try and keep to time where possible. I'll hold up the sign to let you know.
Okay. Hello, everyone. My name is Carolina Cichowska. I come from Italian National Institute of Oceanography and Applied Geophysics and University of Trieste, and I would like to talk about some approaches to model invasions using ecosystem traits and ecological interactions. So as an introduction, we know that marine biological invasions are one of the biggest threats to marine biodiversity worldwide. They cause a lot of environmental problems, social and economic impacts, and the Mediterranean Sea is a hotspot of marine invasions. We have species extending ranges natively within the Mediterranean, but also coming from the Atlantic and from the Indo-Pacific. Most of them are coming from the Indo-Pacific, and just looking, for example, at fish, our collaborators' database ORMEF of exotic fish species have recorded 221 species so far. So it is a big problem, and it's recognized. And so far, marine invasions have been studied with food web models, which is great because we can create these synthetic experiments to look at invasions, impacts on food web, but it's a little bit tricky to incorporate them, to model them in, for example, Ecopath. There has been different ways tried, for example, using two static models, pre-invasion, after invasion, but also the temporal simulations. For example, using tricks like forcing the biomasses or forcing vulnerability function. And here I would like to present some other approach using these environmental ecosystem traits and ecological interactions. So what we've done here, we did synthetic experiments of an invasion in Adriatic Sea as a case study, using ecosystem traits and ecological interactions such as potential invasive alien species, native predators and competitors declines, behavioral traits of predators, and vulnerability changes to the coming invader. We specifically wanted to answer in this presentation three questions. What are the ecological conditions for which invasion is successful? And what is the role of residential predators, competitors in hampering or facilitating the invasion? And are there differences in maximum established biomasses that invasive species can achieve? So to do that, we used two. So the case study for this approach was the Adriatic Sea. We used two different Ecopath models of Adriatic Sea. One of Marta Koll, North Central Adriatic from 1990s, 38 living groups, and the other OGS model of Adriatic from 2020s with 73 living groups. So we use these two different Ecopath models to have different taxonomic resolutions and to see how our invaders perform in both. So the invasive species that we used was Fistularia commersoni. We chose this species because of ample information on its biomass and on its life history traits and other things that we needed for Ecopath, because as you know, probably there is really limited data on invasive species. We inputted it with very low biomasses as a propagule to the model, and we ran the simulations for 50 years. We did several settings, and these generated 95 different types of scenarios for us. Oh, okay. That's not my presentation. That's the previous one. That's okay. Okay, so we wanted to specifically target the interactions between invasive alien species and the prey, and the predators of them and the competitors. So what we were trying different settings. For example, we were changing the biomasses of the predators or competitors in the ecosystem. We were changing the vulnerabilities, so the strength of the interaction between invasive species and the prey, changing over time, evolving as if the prey is learning about the new predator in the environment following the Langseth method. And also changing different behavioral parameters on the predators. So, for example, we were playing here with the EcoSim behavioral parameters such as maximum relative feeding time to drive the invasion, basically using these ecological interactions. And here I will show two different results. So the first part will be about the invasion trajectories. Specifically, I will talk about some values regarding the invasion biomasses at invasion peaks, rates to reach these biomasses and the established biomasses at the end of our invasive species. And I will look also at some food web effects, so biomass decreases, increases of different functional groups in our models. Okay, so here we have two different models that we use with two different taxonomic resolutions. And on the x-axis, we have the rates of invasion, so slow invasion, fast invasion. On the y-axis, the biomasses at peak, so low and sorry. Well, yes, please. Thank you.
Sorry. While they're doing that, if you have anything on your presentation that is not just PowerPoint, you might want to check it before you speak. If you sent us an update, you might want to check it before you speak.
It was this one, yes. Yes, yes, this one, this one. Okay, 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 okay. So basically, so the rates of invasion, the biomasses, so every circle represents different scenario. And we can roughly see that basically two different, the models of the same Adriatic Sea, but with two different resolutions, they show similar trends for our scenarios. So basically we can divide the invasions into these four different groups: the big biomass invasions, the fast invasion, medium pace, and slow. I will focus here on, just for simplicity, on the OGS model, on some of these groups. So how they were driven, these invasions. So what was facilitating this, for example, big biomass invasions? And here we can see that the very important interactions were between the prey and the predators, the invasive species and the predators, the invasive species and prey. So these big biomasses invasions were possible when we had, for example, predators in decline, when the invasive species was adopting the predator avoidance strategies. Similar situations, so again, these very important interactions for facilitating invasion were between invasive species and predators, invasive species and prey for fast invasions. This time, for example, invasive species it was facilitated to increase their abundances very quickly when there were decline of predators, when the vulnerabilities of prey to invasive species were evolving over time, but also when the feeding time of the invasive species was constant. So it was kind of a bold predator. And then we have some results regarding the food web effects. So here the rows, they represent the different scenarios. And the columns, they represent different functional groups in our model. So we have the prey, the competitors, the predators, and we have the other functional groups of the food web. Red, the biomasses decreases, biomasses increases in blue. And again here, so generally what we can roughly see, so it's one model, the same invasive species. We are still trying to drive the invasion using the ecosystem traits, the interactions between the species. And already here we can see that the effects on the food web, they differ a lot. So there are some commonalities, for example, like the invasive species prey is declining over many scenarios. Then later in the food web as well, there are some commonalities like increases of biomasses, decreases, but we can see there are also a lot of differences between the scenarios, especially, for example, when we change the behavioral strategies of invasive species, or how we also have driven the invasion. So it's very important here to think before we are actually simulating the invasion, if we are interested in the food web effects, to think well through what drivers and behavioral strategies we are using. Yes. Thank you. So just in conclusions, to answer these three questions here, what are the ecological conditions for which invasion is successful? So we found that, sorry, I'm out of breath. increasing vulnerability of native prey to invasive species over time. So following this Lancet method is very, very, very useful. The decline of recipient predators is very important for the invasion to be successful. Of course, we have to take into consideration that this study was just one case study with one type of invasive species. But we found here that, yes, the role of predators were more important for facilitating and hampering the invasions, and competitors' role came as second. Are there differences in maximum established biomasses an invasive species can achieve? From our scenarios, it looked like yes, there's a ceiling that most invasion scenarios reach, and to go above the ceiling, several settings have to be met. The increasing vulnerability of native prey over time, the predator decline, and invasive species predator avoidance strategies. But on the other hand, the rates of the invasion, for example, are the fastest when we put the no predator avoidance strategy for invasive species. Okay, thank you. Thank you.
Has anyone got any questions? Yes.
Thank you for your presentation. I have a question about the construction of the model. How did you define the predator of the invasive species? And how did you define also the proportion of the invasive species in the diet matrix of the predators?
In the diets of the
Yes.
predators. Yes, exactly. So definitely, that's a very good question. So basically, we could find, for example, a lot of information for this invasive species for its prey already for the diet. For the predator, so who is eating the invasive species, it was trickier. So I found it in several different publications that, yes, there were in the stomach context analysis, there was the Fistularia commersoni. And basically from the literature, we tried to, yes, to put it together, but it was, yes, it's quite limited, of course, because as well for the Adriatic case study, for example, it's not that there is so many Fistularia now there of our invasive species. So, for example, for this paper, I had to look more in the Eastern Mediterranean. So, yes, like this. Sorry, did I answer your question?
Yeah, so yeah, it's okay. So you looked at another ecosystem and you made the same choice.
Yeah. Yes. Yeah. Yes. Unfortunately, yes.
Okay, I know it's not easy to.
Yeah.
Questions? I was just wondering if you looked at the kind of socioeconomic impacts or whether it's the next stage to look at fisheries and if this poses different opportunities to fisheries.
Yes, thank you. This is actually our second part of the project about the fisheries impact and how we can use these different invasive species. So yes, we had some preliminary views, but it's still not going to be published. So I don't know if to. No, it's not a sin. No, but there are still not many results. Yes, but that's definitely the next step. Yes.
Thank you very much.
Thank you.
There's no link, is there? Is that what you're saying?
But I don't know if they're here.
No, they're here?
Oh.
Oh,
But you don't have the presentation? You have no slides?
Yes, I have. I did send.
Well, we don't have them.
I'm sure it's here.
Yeah. Okay.
brilliant. Oh, welcome to the stage.
What is it?
All right, take it away.
Thank you very much. My name is Morteza Salahi, and I'm a postdoc in Alfred Wegener Institute. My project is modeling the future chains in Dogger Bank food web, and I work in the Warren-C station on Zelt. So energy policy in the European level is going to have a climate neutrality by the year 2015. 50 and fight biodiversity loss and pollution. So member states have to come with their policy plan for the future, and constantly there is assessment and development sustainability measures. The plan for having this ambitious level of the clean energy, as we say, like produced with the offshore wind farm is 340 gigawatts by the year 2050. But right now is the plan and how to measure the environmental impact is maritime spatial planning, and we saw that there is this gap and to understand ecological aspects, and we propose this project for ecosystem-based assessment and management purposes. So here's some statistics about these plans for the future energy production and of the offshore wind farm. As you see at the top, the second one, the North Sea, it's a very huge number. And in specifically in German part, there is a yearly increase of the 12% by year, each year. As you see here, the impact landscape of the German EEZ, as you see here in the map. My project is at the top, you see over there, is Dogger Bank, which is also an MPA. It's a marine protected area, but it's proposed to be a protected area, but not, maybe in the coming years will be approved. And you see there is some places with the wind farm, offshore wind farms planned for the future, the gray ones, for example, and the green ones are already constructed and operating. So ecosystem of the Dogger Bank is how it it's created by the glacial process and then submerged by the sea level rise. So it's very shallow area and have these sandy sediments. Also, during winter, there is a high productivity there because of the circulation of the water around this Dogger Bank. And also we have in the German EEZ, you see over there, we have 3 marine protected area that my other colleagues might be in the audience working on the others, but my project is on the Dogger Bank. And here you see a picture of our beautiful station. What are the impacts or the potential impacts of the offshore wind farms? Are closure and avoidance, as we say, of the fisheries in that area of the wind farms. What are the impacts are? The other ones are noise pollution, especially affecting marine mammals, and electromagnetic fields, for example, for sharks. And we have this huge impact on the birds, seabirds, also in the 3 approach for population level vulnerability and collision. This differs for the different species. For example, for gulls, most likely they collide with these wind farms, and for guillemots, they might be displaced. So what happens after we install a wind turbine in Wadden Sea, and North Sea? It first affects the sediment. So when it's can you please not? There is my screen is disrupting. Yep, thank you very much. First, what happens is the sediment type changed. Sediment type from sandy sediment, and it became more fine sediment. And then after that, polychaetes comes, and first there are filter feeder polychaetes, for example, come around, and then after that, predatory polychaetes come around for the first 2 years, and then for the third year, it depends on what type, because there is this vertical spectrum of the species. For example, for the intertidal level, there is barnacles dominated, and then as we go down, dominated by the blue mussels for the third year, and then associated species of the blue mussels come around. And also sponges also in the bottom, and sea stars increase. I'm sorry. Does it change anything? Okay. No.
We're having some technical issues.
What happened? To hide that?
And now it's working.
Is
Test
it
that.
working? The magic.
Yeah.
And then in the fourth year, the fish predators are the dominated species for the mussels, blue mussels, for example. And then how we did approach to this modeling, and we did a setup for it. First of all, we did the baseline for the pre-installation of the wind farms in the place. For the base model, we aggregate all the data and all the food web in the Ecopath, and then we build up our Ecopath for the pre-installation food web. And then for the after post-installation food web, we came up with this idea to have these multiple snapshots of the year for each year and go forward and apply the changes and kind of have a circle and go back again and do another snapshot of the model, and then measure the convergence. If it's achieved, then we have our post-installation model. This is the full web of the pre-installation of the wind farm in the Dogger Bank. As you see, we have fish species in the middle, and we have fisheries over there. We have marine mammals, we have birds in the species level, which is pretty decent. And then we have benthic at species level as well. And here you see it's Dogger Bank, although it's a sandy sediment in the pre-installation model, it's still very much, you see here the big number of the detritus flow to the system. And here we have a pre-installation against here also post-installation model as well. As you can see the changes, how benthic, you can compare the benthic species, how they change, and we exclude the fisheries and then change the other, and then you can see the changes of the other species as well. And here I just want to thank all my lovely colleagues and happy to have questions.
Thank you very much. Do we have any questions?
So this is a dark history in my lab, but my advisor had a wind farm paper that has been rejected 3 times. And here is the problem. So if you want to use Ecopath with ecosystem to model wind farm, usually you have to be a closed ecosystem or closed system. And if it is a closed system, it is usually too big. And then wind farm is a very small part of it. So it is very difficult to say wind farm has any effect. And if you only focus on wind farm, that wind farm area, it becomes an open system. Like here, you have birds. Then birds cannot be modeled as a, so I'm wondering, well, maybe you have to address these sooner or later.
Yep, thank you very much. Actually, I think the biggest that biggest thing that we are proud of is this data that we have. It's for example, for birds, we have really species level data for it. So it's not like simulation or anything. So for each year, we have these, even for some species for months, we have these real data. So I think for at least for most of these species are reliable.
Question?
Thank you for your presentation. What parameters do you change when you create the next the food web of the next year? Production, biomass, which biomass?
Basically everything. We start for each year, we start from scratch. We find out what species we are dealing with, how would change the parameters. We study all the literature to see how it can affect the parameters. Basically, we do multiple snapshots separately and then measure the convergence, it's happening or not.
Okay, thank you.
Okay, we'll move on. Thanks very much. I think as well, there's quite a lot of people in this room that are developing offshore wind farm models. And I think EWE has probably been recognized as one of the best tools to deal with it in terms of its cumulative impact. So I think maybe there's room for discussion there later. I think we've got some talks on Wednesday about that too. But moving on, he needs no introduction. All the way from Scotland. Kieran, come to the stage. First talk, modeling the effects of offshore wind. Oh, that's timely.
So yeah, we swapped chairing duties so that Jacob could introduce me for a talk that also happens to have his name on it as well, because that's how we do things in Scotland. And Natalia, who's also in the room somewhere, I see you, also named on this. So so this is a wee project where we're looking at the effects of offshore wind farm development in ecospace, predominantly to look at localized effects from the development. The area that we're considering is the west coast of Scotland environment, an area that Ecopath EcoSim's been utilised extensively. Quite recently, Serpetti et al. Developed a high-resolution ecospace model for sort of like a sub-region within the West of Scotland environment. So they developed a high-resolution ecospace map which incorporated the Isle of Muck, a small Scottish island, and the surrounding marine environment. So this is a 100 by 100 meter resolution benthic habitat map. And the purpose for that was to study the effects of a hypothetical multi-purpose platform that included a wind turbine as well as a fish farm. So here we're going to utilize we are using the same ecospace map for a larger hypothetical wind farm development. The reason for that is because in a different part of the west of Scotland, a lease agreement has been made for a relatively large wind farm development, which will probably include about 100 turbines, which are likely to have large fixed jacket style foundations. And so the idea for this project in general is to take an approach at this smaller ecospace map, this high resolution map, and then eventually apply the same approach to a different area. It will be a larger ecospace map, but at the same resolution. And if we get there further down the line to then replicate this for another ecospace model in the North Sea. So we're utilising the West of Scotland Ecopath model, which contains 43 functional groups and has previously been fitted to time series and EcoSim for the period 1984 to 2013, and work is or will be ongoing to fit it up towards the present day or close to the present day. The ecospace map that we have, so we're repurposing the work that Natalia did. And so we're incorporating a new habitat type in here called turbine foundations. Yeah. And so this is in a map which already contains a series of different sediment benthic habitat types at different depths. So we're putting in 14 turbine foundations at an area to the northern portion of the map. So this is an area that's predominantly mixed sediments and muds at a depth of 50 to about 200 meters. A single foundation in our model takes up one cell, so a 100 by 100 meter grid cell. And we're introducing these turbine foundations mid-simulation using the spatial temporal framework. We have a series of different responses in the model to having this new habitat type in there. So in the original Ecospace model, the Isle of Muck model, it included noise effects from single turbines on marine mammals and partial dislocation for seabirds. So we're including those, but on a larger scale because we have more turbines. And the nice thing with doing it this way and having these response functions set like this, it means that we can potentially limit the time that those responses are occurring on. So maybe marine mammals get used to the fact that there are turbines there and start utilizing it again. Well, we haven't done that yet. Preferential habitat foraging usage was used primarily for other functional groups, how they would utilize this turbine foundation habitat. And we had to consider several different scenarios for this. So where we know that species have been observed at turbine foundations, are they just as likely to forage here as in other preferential habitats? Or are they actually more likely to utilize this type of habitat? So we've considered these different types of scenarios. As a result of only having a modest response for epifauna, having a preferred usage of turbine foundation habitat, we ended up changing how the model calculated biomass for epifauna and forced the biomass to up to 36 times that was predicted. And that's based on observed data from the North Sea. So I'm just going to present a few different scenarios. So it's a sort of cumulative effect of introducing something in additional to the prior scenario. So we have a baseline where we have no wind farm development. Scenario 1 includes sort of like a base usage for functional groups using the turbine foundation habitat. We're including this forced increase in epifauna biomass and this reduced seabird biomass by about 45%. Scenario 2 then adds in a noise effect for marine mammals and is effectively causing them to be displaced from this area. We're then restricting fishing in scenario 3. We're increasing the usage of the habitat in scenario 4. And scenario 5, we're just taking this at another level again. So saying that, for example, our immature functional groups, or immature fish functional groups, will potentially utilize this habitat 5 times more for foraging compared to others, double for other fish. So we get series of different outputs from EcoSpace. I'm just kind of utilizing this figure to indicate. So if we draw a region around about where our turbine foundations are located in the map, so we're just looking at the output for this area, and we can clearly see where the foundations are because we've got this really high forced biomass for epifauna in this area. So this is just a table showcasing percentage change in biomass based on baselines across these different scenarios for some, well, I deemed interesting functional groups or functional groups that had interesting responses to them. At the bottom, we have this consistent increase in epifauna biomass. And that was actually one of the drivers and the funding for this project was, well, what is the effect of having significantly more epifauna in an area? So under scenario 1, we see a modest increase for our fish functional groups, maybe slightly higher for flatfish. We see a relatively significant increase in biomass for lobsters and edible crab. That's mostly in relation to actually providing them with a habitat, which in an area which they wouldn't maybe typically be otherwise utilizing. The biggest change that we see for our fish functional groups is by removing predators, or not completely removing, but mostly removing predators from that area in terms of marine mammals. So maybe not hugely surprising. And this doesn't really have much of an effect on our crustacean groups shown here. By restricting fishing, we see again another very slight modest increase in biomass across different functional groups as a response to that, but nothing really significant. It's worth keeping in mind that we've got a really low kind of small grid cells, and for example, our fish are going to be able to move out with this region and out with these grid cells within a time step quite easily because of the dispersion parameters. Scenario 4, when we start increasing the usage of turbine foundation habitat, we start to see some interesting changes occurring in biomasses for some group, and that's replicated when we go to that scenario 5. And what we're seeing is predator-prey dynamics, because we're effectively forcing or encouraging groups to occupy the same habitat where one may be the predator of another. So a nice example of that here is why we see this reduction in Norway pout and this big increase occurring in saithe. Saithe eat Norway pout. So if we tell them that they're living in the same place, Norway pout aren't going to be too happy. Saithe are going to be very happy. And of course, scale is interesting, or a must kind of consideration in this as well. So we're already assuming that this larger West of Scotland Ecopath model represents this much smaller area. But then when we're looking at these regions, so a 14-kilometre square area that incorporates all our wind turbines in this scenario, or a smaller area which surrounds just a single wind turbine, we see a series of differences depending on the functional group that you're considering. And this is something that the funder is particularly interested in this project, given that they're interested in looking at marine net gain. I was ignoring them. What they're interested in is the potential for marine net gain at wind turbine development sites. So just to say that there's ongoing work where we're developing a larger Ecospace map at the same resolution at a different location. And we've also got a proposal, and I've got a proposal in to look at a North Sea site, and where we're going to potentially also look at doing a lot of analytical work and stable isotopes. So there's definitely people here I want to talk to about that. And just some sort of general conclusions before Jacob and Sheila throw me off. So redeveloping existing models to investigate new questions can be a relatively fast and efficient method. The model extent's always important. It can be a limitation, but forces additional assumptions to be made. Ecospace is obviously a powerful tool that can be used when considering the impacts of structures and potential mitigation, and the spatial temporal framework has clearly got a role in there. It's really important, particularly in Ecospace, to be considering different scenarios, the different effects, the combinations of these, especially when data is limited or only just emerging. Happy to take any questions. If there's time.
I have time for one quick question. Last question. Sheila?
Can I be nasty? Oh, no, Marta hasn't had a question. Marta, we let you ask.
I'll be nice. I just had a question about the scale, right? You have this 100 by 100 meters, which I think is great. Why did you go to so high resolution? Could not make it work with a lower resolution?
So the interest was in terms of looking at the artificial reef effect in Ecospace from having these turbine foundations, and ideally you would potentially want to go to a lower resolution, a higher resolution again, because the reef effect is going to be so localized. You're only looking at a tiny part in the area. That's what the funder is interested in, is saying that, well, you know, if we draw a circle around about where our development is, we want to know what the specific changes are in that area. We don't really care what's happening out with it, especially given the Crown Estate and the UK are involved, and they're the people who are providing the leases for these wind turbine developments, wind farm developments.
Yeah, but if you try to express it as a proportion of the cell,
yeah,
would you get the same, or have you tried that?
No, I actually haven't, because I've just mostly been working with the high resolution model that we already have. So I've not done that. I think it would be interesting to kind of do that and say, okay, in a larger grid cell, and we say that only.
I think you would make our lives easier if you show that doesn't matter. You can go a bit lower resolution and you get nice results. Please.
That Leah had done the hard work in creating it, so it was worth just utilizing the work that she'd already done.
I asked that question, answer that question. On my model, I was looking also at salmon cage associated to the wind farm, and there was a particle tracking model. Then it was modeling the eutrophication of the footprint of the wind farm on the aqua farm, salmon farm. So the high resolution, it was due to the high resolution of the Lagrangian model. Then it was drawing the enrichment on the seabed from the salmon cage.
But still an answer. Maybe we get similar.
Yeah. Thank you.
Okay, up next, we have Darcy with a talk modeling ecosystems and fisheries. modeling ecosystem fisheries of the Vanuatu archipelago, Mozambique, using Ecopath with Ecosim.
Just going to quickly check that this is the right presentation. Fine. They're minimally different anyway. Okay. Hello, I'm Darcy Anderson. I'm a master's student at the University of Cape Town, and my project is looking at the Bazaruto archipelago in Mozambique, which is like a low data environment, and developing methods to model that. So, Bazaruto is an archipelago in southern Mozambique comprised of 5 islands, and together with the San Sebastian Peninsula to the south, there's about 10,000 people that live in the area, most of whom rely on fisheries for both income and food security. This is somewhat concerning because Bazaruto is in the Western Indian Ocean, which is the fastest warming non-polar region globally. So there is the potential that fisheries will decline due to climate change. So that's a big impetus behind modeling this area. So in terms of the ecosystems that make up Bazaruto, one of the most influential are the seagrass. They're very extensive and they're home to about 160 fish species, approximately half of which use these as a juvenile habitat before ontogenetically migrating over to the reefs. It's also a very important habitat for the critically endangered dugong population in Bazaruto. The reefs are very diverse in terms of their morphology, but the thing that unites them is that they are considered some of the healthiest reefs in East Africa, and this is largely attributed to them having been protected since 1971, when the Bazaruto Archipelago National Park was established. And in terms of area, sand is by far the dominant habitat, which has quite a low density fish community. There's also quite a significant mangrove forest around San Sebastian that also is used as a juvenile habitat for many species, and as well as significant pelagic influence, as Bazaruto is very close to the continental shelf edge, and that brings in billfishes and large fish like tuna. So in terms of the fisheries, the artisanal fisheries operated from dhows and rowboats using seine nets and hand lines are the most important fisheries. These primarily occur on the seagrass beds, and the majority of catches are of juvenile fish, which is why it's important to understand those linkages. And then there's the issue that the majority of fishes, over 90%, have reported declines in fish size and abundance between 2006 and 2016. And then additionally, there's a gleaning fishery, which is a subsistence fishery of invertebrates, as well as a sports fishery, primarily for black marlin. So in terms of the functional groups we have going on, a lot of them are quite aggregated, but I've separated out the more important fishery species, that being the sardines, the scads, and the silver biddies, as well as the dugongs due to their importance to the national park. So the main issue is data gaps. For Mozambique, Bazaruto has had a lot of research done on it. For ecosystem models, Bazaruto has had very little work done on it. And so we're lacking a lot of the key data. And so my challenge is to come up with how to address those. So I'll get more into this on the next slide, but we're developing a satellite-based method to reconstruct fish time series so that we can fit them, the model, and run climate simulations. To address our total lack of phytoplankton, biomass, and productivity data in the region, we're planning on utilizing ocean models that cover the area and extracting primary productivity parameters from those. Very luckily, we have found great collaborators in other researchers that have previously conducted work in Bazaruto who've shared their data, and that will enable us to calculate some of the biomass data from what they've collected. And then also my project fits within a larger project called Remotab that has a lot of people currently conducting research in Bazaruto. And as more data comes out from those projects over the next few years, those will also be incorporated into the model. And then finally, we'll be building on existing work to reconstruct the seagrass meadows and the mangrove biomasses using satellite data. So in terms of the fisheries reconstruction, the first stage is to actually access the satellite data, which is very difficult, but we'll be using WorldView 2 and 3 imagery because it has very fine resolution. And then building on the work of McCarthy and colleagues, we'll be running an algorithmic detection algorithm, to detect the boats from the satellite imagery, which has about an 86% accuracy rate for detecting dhows. From that, we'll calculate effort, taking that as the average number of boats at sea in Bazaruto per day. Then we will use an open source fisheries database called WeoFish that exists for all the countries in the Western Indian Ocean that has lots of data on the on artisanal fisheries that has been aggregated from governments around the Western Indian Ocean and their per country basis. So the thing we're interested in is this catch rate of kilograms per boat per day caught. And we can combine that from our effort data to calculate catches. Then we are also working on getting access to the fisheries data that has been collected in Bazaruto since the end of 2021. And that needs a fair bit of work to get it into the same format as our reconstructions. But once they are, we can then calibrate those reconstructions and get an idea of if the reconstruction has any basis in reality. And then go into the field and conduct some ground truthing experiments to ensure that what we're inferring from the satellite imagery is reflective of the reality on the ground. And then the final step is to repeat this many, many times to create an actual time series. So in terms of what I've got done to date, I'm about 70% of the way through my parameterization and just working with the data and getting new data all the time to build the Ecopath. So over the next few months, I'm going to be parameterizing and balancing my Ecopath model. And one of the parts of that will be using multi-stanza groups to represent the species which ontogenetically migrate from the seagrass onto the reef, as this is a very important part of this system. Then we'll be moving on to the ecospace and developing that until February. And then come June next year, hopefully, I will be starting my PhD, which will begin with conducting the satellite-based fisheries reconstructions, which I understand it's very odd to do an ecosim after an ecospace. However, because we are trying to get the WorldView data from the European Space Agency, this takes a lot of time and it's a big process. And so just to meet those constraints, we're having to flip things about a bit. After I've reconstructed them, I will then finally be able to build my ecosim model. And the things we're looking at that is running simulations of our catches under no climate change and moderate climate change, and then a high scenario, and then also assess tipping points within the ecosystem, primarily of the seagrass and the reefs because of their importance. And then also, based on that, revise the ecospace model so that then we can run spatial temporal simulations. During my PhD, the goal is also to work with Sam and work on other ecosystem models within the Western Indian Ocean. Which brings me on to the final point, which is even though there is very important applications of an ecosystem model within Bazaruto, the goal of this project fundamentally is to be a pilot study for a larger project that we're calling WIO 2100. The goal of which is to understand how climate change may impact the ecosystems and fisheries in the Western Indian Ocean between now and 2100. So there's a very big need for this. The Western Indian Ocean, countries in East Africa, communities there have a very high reliance on ocean resources for food security and for livelihoods. It is, as previously mentioned, also the fastest warming, heavily populated area on Earth. And also it's very understudied. And so far, there's been limited modeling in the region. So our models are going to be the Agulhas Current, which is from Mossel Bay up to the coast, the border of Mozambique and South Africa, and then the Mozambique Channel, and then the East Africa Coastal Current, which incorporates Tanzania and Kenya. We would like to do Somalia, but the data challenges there are far too significant to overcome. So yeah, thank you for giving me the time to speak today. And
Do you have any questions?
Thank you. Really nice presentation, Darcy. More a comment rather than a question. It'd be really interesting to see the differences that you get with your catch data based on the effort and what you get from satellite data and what has previously been reported and the new observed data that you're getting. So recently been involved in creating an ecosystem model, Ecopath model for Mauritius.
Okay.
And knowing the amount of unreported fishing that goes on there, which probably might even make up the majority in terms of inshore fisheries is unreported fishing. So that'll be a really just an interesting exercise in itself,
Yeah.
I think.
No, the park is also interested to see what numbers we get for the satellite data, because they're not 100% sure of how closely their fisheries data, how much is going unreported. Yeah.
Hi.
Hello.
You mentioned talking to people about the artisanal fisheries and how they've noticed a drop in catch.
Yes.
Have you considered other ways to incorporate their information with the artisanal fishers and how to keep their information in the modeling going forward?
So a lot of the information of the fisheries is based on a thesis done by Chiara D'Agata in 2016. And she went and did structured interviews with fishers in Bazaruja Island, in the San Sebastian Peninsula, and also in Vilanculos, which is the kind of town on the mainland close by. And so she did that, and then also another researcher, Linda Egerton, has been very generous in sharing traditional ecological knowledge data with us that will inform the ontogenetic migration stuff. Yeah. Yeah.
Do you know of the work done by Jennifer Rearan? She was one of the last seminars she also dealt with data poor situations in Mozambique. She
No, I
wrote a nice
haven't.
paper about it. We'll send it to you, right?
Okay, thank you so much.
There's no hat. I don't know if there's supposed to be a hat next to Darcy. Yeah. Yep. Okay. Okay. There's no hat here, but I guess there should have been a symbol for Darcy as well. Yes. We have another student up now. We have Eduardo talking about searching for ecosystem modeling realism when apex predators help each other.
Can I check? Because I said it too. Can I check if it's the new presentation? Yes. Ah, it's correct. It's correct. Thank you. Yeah. Hello, everyone. My name is Eduardo, and I'll be talking a bit about our search for ecosystemic modeling realism more specifically about a situation where apex predators help each other to catch the same prey in a facilitative interspecific interaction. So just a little bit about our study site, which is in the map over there. The Laguna Estuarine system is formed by 3 coastal lagoons, which are connected to the Atlantic Ocean by a small inlet right there at the red stars. And in Laguna, we have something that we call the cooperative fishery. So the cooperative fishery is an interaction that happens between dolphins and cast net fisher folks that cooperate to catch mullets. And in Laguna, it is considered to be a mutualistic relationship. There are a few other places around the world where happens something like that, which is 2 more places, one in South Brazil as well, and other places around the world, which, well, there's something like that happens, but are not considered as truly mutualistic interactions. And in Laguna, it is socio-ecologically very important. It is considered to be natural and cultural heritage. And by now we have a multidisciplinary group that have been studying several aspects of the cooperation, such as the behavior of both dolphins and the fisher folks, population parameters of both dolphins and the fishermen, and the bioeconomics of the fishery. And right now for my PhD thesis, we would like to understand the role of the cooperation on the food web and what's the impact of the cooperation on the ecosystem. And nowadays, the cooperation is threatened by the industrial overfishing of mullets and also by the bycatch of dolphins by the fishery of gill nets, which kills almost 1 individual every year. So a little bit more about our system. It's also very interesting ecological place because it's considered to be a transitional climatic zone. So it's the austral limit of mangroves in South America. So southwards of Laguna, the mangroves are replaced by salt marshes, and northwards of Laguna, the salt marshes are replaced by mangroves. And these are both very important fishing habitats, right? Because they are nursery grounds, and they are nursery grounds for several species, several important commercial fish species. And the Laguna Estuarine system is also a very important fishing ground for the state, for the whole region. Almost half of the catches of pink shrimp happens there. And also we have significant catches of many fish species such as catfish, flatfish, croakers, mullets, and blue crabs. So putting all together, it's imperative that we implement ecosystem-based fisheries management, making the Laguna Estuarine system a suitable candidate for an Ecopath model. So here's it's supposed to be the cooperative fish in action. It's a GIF. I don't know if it's going to work. Just a second. Oh, can I try again? Sorry. It's this one.
This one, no?
This one.
Yeah.
Yeah. Yeah. It's. Yeah. All right. Okay, so as you can see, there's the dolphins. They locate the fish schools, the schools of mullets, and they push the mullets towards the channel wall where the fishes are standing. So the cast net fishers, they understand that a few signals that the dolphins will make. There is a repertoire of signals that the dolphins will make, and the fisher folks will throw their nets. I'd like to show it again, but well, maybe next time. So what's the goal here? What's the challenge for us? It's to implement the cooperative fishery in EWE, in an Ecopath model. So can we include individual behavior in an Ecopath model, and what are the food web effects? So these are our main questions as we hypothesize that yes, there will be an ecosystemic impact of this individual behavior that dolphins make. So by now, our model is a work in progress. I just would like to highlight that we've been dealing with 6 fish groups that are the most important fish resources with 2 stanzas. So we have other fish groups that are not as important. And we have been dealing with both the cooperative and the non-cooperative components. So both for dolphins and for the cast nets, we have cooperative dolphins, non-cooperative dolphins, and cooperative cast nets and non-cooperative cast nets. Yep. Okay, so just a little bit about the trophic ecology of dolphins. As you can see, the mullets are the most important item in their diets. And it's a challenge for the near future to differentiate between the diets of both cooperative and non-cooperative dolphins. We really hope to do that in the near future. And a few numbers. So regarding the cooperative fishery, we have 21 dolphins, but as you can see, they catch much more mullets than the non-cooperative cast nets, which are the non-cooperative dolphins. And well, we have lots of mullets being caught in the system throughout the year, and 13 are from the cooperative cast nets. So that's a lot. And well, what are the possible solutions for our challenge? We could simply compare the models with and without the cooperative components, or we could try to adapt the mediating function in Ecopath to include fisheries groups as well, fleets. And we have other mathematical possibilities. That's all very initial, very incipient. So it's to be further developed, but we could try to implement a facilitation matrix. And as it was suggested, we could also try to create a relationship between the effort of the cooperative cast net that will make the mullets more available for cooperative dolphins. And by doing that, we expect to understand the role of individual behavior on the food web, that if we could implement something like that in an Ecopath model, it could also be helpful for another systems around the world that have human and wildlife facilitative interactions as well. And well, thinking about operationalization of Ecopath, we really hope that by implementing that on our system, we could have better dialogues with the castnet fisher folks, which are the main stakeholders maybe in the system, and therefore preserve a cultural and natural heritage. I'd like to say thanks to the event organization, to the VLIS for the supporting and for your attention. Thanks.
Hi, thank you for your talk. I have more biological questions. I don't know if I missed it, but what is the benefit for the dolphins to do the cooperative fishing? Because I see it for the fishers, but what is the benefit for the dolphins?
So there is the confusion effect. So this is something that happens for a while now. It's not something new. It's not something sporadic. The same dolphins, they always come back to the same places to cooperate with the same men. And well, we have some hypotheses from another papers that when the cast net is thrown in the water, it's easier for the dolphins to catch the mullets that try to swim away from the ones that are being caught by the fisher folks. So that's basically it. But we don't actually have footage underwater because our water is very dark. So we still don't have 100%. We are not 100% sure of that.
Yeah. Oh, there's another question. Go with the other one.
Oh, I was just wondering, great talk, by the way. I was just wondering if you think that the strength of that relationship is density dependent. So what happens when there's less mullet? Well, there'll be less of an incentive for the dolphins to actually cooperate.
Yeah, that's a great question. It's something that we'd like to know as well. There are some hypotheses. It could be stronger or weaker, right? If there are less mullets. So it's something that we actually would like to try to experiment in Ecopath as well. But it could be split in two ways. It could be stronger because there is less mullets and they will need more this type of feeding habit, or it will be a weaker relationship. We don't know.
Should we go Sheila, Billy, then Dave, or I don't know.
Oh, it's okay. Mine is, yeah.
Really interesting talk. And I have a question, and I just want to get your opinion on it, because we sort of had the opposite situation in the Gulf of Mexico, to where the interaction between dolphins and fishermen is more antagonistic. And so what happens is, as the fisherman is reeling in a fish, the dolphin comes by and takes it off the line. And the same issue with sharks. And so if you ask anybody in the Gulf of Mexico, depredation by, or any recreational fisherman in the Gulf of Mexico, depredation by sharks and dolphins is like the biggest issue. So I've sort of stayed away from that question because I could never figure out how to model it in EcoSim. So I'm wondering, based on your experience, or Ecospace, and I'm wondering, based on your experience with this cooperative relationship, do you think that we could model sort of the opposite effect to where it's more of an antagonistic relationship between the predator and the fisherman?
I mean, in my opinion, by now, what we can do in Ecopath is that, right, to consider them both as competitors. So, but that's not what we want, because we consider them not to be competitors. We consider them to be mutualistics. I don't know if I answered your question.
Well, think of that. I just want to give you the put that thought in your mind that
okay.
This this process plays out in the opposite effect in other regions, but I think you figuring out how to do it this way, we could probably learn how to model the opposite.
Okay, thanks.
Yeah, it's not a question. I had some discussion with Eduardo when I saw his abstract, and we talked about how to implement what he was looking for, this mutualism into mediation. And there is a rather simple way of doing it, but it calls for some programming. And it really reflects a problem we've had in recent years, which is we don't have any, what you call it, pool of programmers that we can just pass this on to. We're going to be discussing that in the What's Next session on Wednesday, because there is so much more that we can do, but we are limited by the number of programmers. We haven't been able to clone Jerome recently. I mean, he's got babies and all of that, but it doesn't help in the short run. So no, this is really one of the topics for the Wednesday afternoon session, I think it is, but one not next is, how do we change this? Because there's so many things going on now that we really need much more effort on the development side. And we need to diversify it. We need to bring it out to more institutions than now.
Thank you.
Okay, our penultimate talk is from Natalia Sapetti, exploring species distributions, ecosystem indicators, and microplastic risks across time and space in the Black Sea ecosystem.
I'm glad you read the title, so I don't have to do it. When I sent the abstract, I didn't know how much time I had, and I didn't know which session I would end up. So I'm gonna fly over the initial of the presentation because it's new ideas, so we are concentrate on the end of the talk. So I developed a new model of the Black Sea for JRC, where we ask, because Black Sea faces a multitude of human-induced pressures, eutrophication, pollution, overexploitation for fisheries, species invasion, and climate change, of course. So I created this new model for the Black Sea, then follow the same structure of the Mediterranean models created by Chiara Pirotti in the past, and she presented the last conference. And one reason is because we want to use both models to assess the impact of policy implementation within the Marine Strategy Framework Directive. So in this context, I introduce you the Bluetooth modeling framework that we created a long time ago. So no, it's not we, they created before I arrived. And it's a modeling tool that has been made for exploring the consequences of European management and policy options within the ecosystem. It's a kind of a digital twin of the ocean couple model. Here we have atmospheric forcing, wind, temperature, precipitation models. Then there are input in different models. So land, river, hydrological model, hydrodynamic model, then they are dynamically coupled in a biogeochemical model, then we call low trophic level. We also have a Lagrangian model, then use the hydrodynamics to model the floating plastic pollution in the Mediterranean Sea and in the Black Sea. And all of them are coupled offline with high trophic level models, then it's Ecopath for us. And as you know, we can have fishing forcing as fishing mortality and effort. So this model have 47 functional groups. And as I said, is coupled with a biogeochemical model, GETEM, BISEM, with the low trophic level. So it's coupled with 2 zooplankton groups and 2 phytoplankton groups. And the model at the moment is fitted to time series. The drivers are fishing effort and net primary production for the biogeochemical model. And here you see the fitted with for the stock assessment groups, but I have the fitting for all of them. I have an ecospace model for this. Then again, is coupled with the biogeochemical. So there is the depth distribution with the niche of the favorite environment in depth and spatially distributed the species in the model. Then another driver is bottom oxygen, because the center of the basin is anoxic, as you can see there. So the oxygen is available only on the northwest shelf and in the shallow water, let's say. Temperature, salinity, small phytoplankton, large phytoplankton, small and large zooplankton, and net primary production, all coming from this hydrodynamic model biogeochemical. So when the model runs, we get distributions of species. And here you can see the same group I show for the fitting, which is the one they are stock assessed within the basin. Then I did some indicators as well, some network analysis. I don't have any of it here in this presentation, and some ecosim temporal changing of the trophic level of the community and the diversity. And you can see the declining over time of these indices, even for biodiversity. The spatial indicators, just to show that there is an higher let's see, biodiversity in the shelf where it's less anoxic. So we get the contribution of the biodiversity also on the benthic groups. But because I'm in this session, I want to concentrate on this idea of building up an ecotracer model for modeling the uptake of microplastics into these ecosystems, and a way on doing some calibration of looking at observations. So the first objective of this project was to create a database. Then you look at microplastic concentration uptakes for marine species. And I read lots of paper and on the microplastic stomach contents in many different species. And I didn't exclude anything at the beginning. So I went from benthic invertebrates all the way up to mammals. So I look also at macroplastics, not only micro. Then I did a statistical analysis in which I look at only fish. So I exclude benthic invertebrates because they have different feeding mode. And I also exclude top predator because they feed in macro particles. So this one, it's the statistical analysis being carried out only for fish and only uptaking from microplastics. So I look at GLM for find out if there were any significant occurrences or mean item per individual as a responsive variable in relation to location or habitat preferences. And for habitat preferences, I use the fish base. So using demersal, other neritic or ocean and pelagics. So the results of the analysis showed that there are high significance occurrences in the stomach content and mean items, some higher mean item per individual in the Indian Ocean, in the Mediterranean Sea for mean items, and also in the China Sea for both variables, and in estuary and mangrove wetlands, so coastal ecosystem, let's say, as you expected. So then I built an ecotracer model, and maybe many of you, I don't know who is familiar with ecotracer, but if I'm a fish swimming in a basin, the uptake of microplastic will be directly proportional to the microplastics in the environment. So what you start as an input is knowing how much is the items, microplastic per kilometers in this case. So you have to do some conversion from volumes to space, and also the inflow and the outflow. So all these data are coming from that paper over there. So they are being modeled and validated. The inflow and the number of the initial concentration will be used to calculate the outflow of the basin. And we are assuming that there is no natural decay of microplastic within the time frame we are using, which microplastics stay there forever. So there is not much of decay. It might break down in smaller particles, but it doesn't degrade that quickly. So using these input parameters, we have a stationary steady state equilibrium of the microplastics in the Black Sea models. And that is what a fish has around it. Then based on how much they are uptaking directly from the environment and how much they're uptaking for diet, so for feeding on prey that have been eating plastic themselves, they will start to have plastic in their body. We are not speaking about accumulation in the sense that we are as many as particles are going in, many are excreted. So there is a stationary high or lower concentrations, but there is no an accumulation over time. So the video doesn't work, I checked, and I'm not going to do any dance on copepods uptaking microplastic, but that is what the video was about. So the first group is uptaking microplastic is in my plastic web experiments, is zooplankton. And the data of direct uptake are coming from experiments, tank experiments, where they use fluorescence beads and measure over time how much microplastic get uptaken, and also the excretion rate. So you can follow actually inside the bodies of the poor copepods how much comes in and comes out. So at the steady state, assuming that there is no decay, the natural decay inside the copepods, we have the concentration per unit of biomass of copepods over time. So that is the stationary state for the zooplankton and the larger small. Then we have a fish, then it comes and eat the copepods. So eat the copepods and they eat the plastic within the copepods. So we have, let's see if I put it, then the copepods, the concentration of the microplastic pollution, it moves inside the diet to the next trophic level. And we have also the concentration that has been measured within the group, because I used the stomach content analysis, then I read. So I have, then this concentration over there, it comes from me reading a lot and find a microplastic per individual from a database. And then I scaled up to the entire population. So the ton, a many individual in a ton, and then the entire microplastics within the population. And I used that values to calibrate this equation and to estimate the uptake from the environment, which is what I don't know. I can't put really every fish in a tank and measure it. So there are some of them that has been done also in the experiment, so you can back validate or calibrate, but only for small fish that can live and behave in a natural, let's say, in a confined environment. So using this approach, I basically get to steady states for all the group. You can see the one increasing, it's the detritus that didn't have any concentration, and then from excretion of the fish, it gets the microplastic in the steady state in the sediment. So the second, it was to assess which species or functional group within this model, they have a higher uptake or higher concentration for unit of biomass. So using that system, I found then the primary consumers in the benthic and the pelagic domain are the one have a higher concentration for unit of biomass. So in this case, we have prawns and shrimp, bivalves, gastropods in the benthic or domain, the demersal domain. And we have small pelagic anchovy, sprat, pilcard, that were the highest concentration in the pelagic domain. And that it's important because the primary consumers are the groups, then they are more feed on when in the trophic web. So they will pass their plastic over to higher trophic level. Then we also did some scenarios for the future in which we test different microplastic changing in the environment. So we assume that there was no action, so we are bad, and we are not taking any action for microplastic release that will impact on the inflow on the basin. So we are bad, and we carry on releasing microplastic in the environment. So it's not a constant, it's a constant increase, has been observed in the last 30 years. Then second scenarios, it was we are slightly better. So we reduce the inflow of microplastic in the basin of 10%. And then we keep being good. So it's constant even to the future. And then we are really good and we decline of 50% in relation to the observed. So what happened, you can see then the microplastics goes on in a steady state, is increasing already, is increasing over time. And then if here I show only when we are really good, so 50%. And you can see what is interesting here, then of course the concentration in the species is declining proportionally to what's happened to the environment. But there is a lag of time, so it's not instantly because it depends on the outflow of the basin. Higher is the outflow, quicker is the response. I didn't show you here, but I compare also with the Mediterranean Sea, then they have a different lag on this concentration changing over time. Then I want to do it also in space. EcoTracer, EcoSpace didn't work really well when I started to do this. So I did the concurrence maps instead, where I looked at, in this case, I didn't for every functional group. But here you can see the Shannon index, and that is the probability of floating microplastic from the Lagrangian model that was developed within our groups. So, and of course, there is a higher concurrence in the shallow water where you have high diversity and high microplastics within the food web. So what I want to say only then now we are planning to build it up the Baltic Sea within the same framework of the other two models with the help with Maciek Tomczak. And we are going to run a future scenario in climate change and the policy implementation, MSY, cap of the catches, bycatches, fishing effort and mortality and so on. And we are looking at using the same structure. We can compare the output also for indicators that are so sensitive of the structure of the models. So we are looking at changes of biomasses, catches, diversity, microplastic uptake, and hopefully also economic incomes. And that is why I'm coming to Santiago course to learn more about it. Thank you.
One quick question.
Thank you. That was really interesting.
Oh, here we go.
So, is there a certain concentration where the microplastic becomes harmful for the animals? Did you model that?
No, I didn't. For the excretion rates then I used, and they were coming for reference and experiments, there is no accumulation, so it's just get excreted. However, that also depends of the size of the microplastic we are using it. For instance, for shrimp and prawns, then they have a higher concentration for unit of biomass, is because they struggle to excrete it. So the microplastic, so the prawns have the hepatopancreas on the top, and the microplastic with the fiber especially, they create kind of a ball of plastics that doesn't flow on the digestion system really well, so tend to accumulate more than in other systems. So it will be the same if they are not able to excrete it properly, or the same rate the uptake comes, that is when their stomach can be filled up. And another things which I don't know, but there are only 3 or 4 papers, then look at the contaminant of the pollution, like chemistry associated to the plastic, and actually get into the tissues so they don't get excreted. It will be like a mercury, then it just sitting in the liver, for instance. And there are studies showing that there are some elements of the microplastic, the chemistry elements, they can accumulate in the tissues, but I didn't model it here.
Right, and it's also a concern for people that consume the
There have been some papers, then they said that yes, how much plastic we are eating,
Right.
and how much plastic we are excreting it, but it's very controversial how much is harming. So I'm not gonna say my opinion on this. Thank you.
We have one final talk now from Kieran, modeling ecosystem uptake of radioactive discharges to the Kattegat.
Okay. So sorry that it's me that's keeping you from your beers, and sorry that you're having to hear from me again. 10 years ago, Ecopath 35, I turned up wanting to pollute your models with radioactivity. I'm back again, still with radioactivity. So I'm going to talk to you briefly about a project I've been doing with Maciek, looking at the Kattegat. So this body of water that sits between Sweden and Denmark. There's a nuclear power plant on the Swedish coast here, Ringhals, and it discharges radiocarbon into the marine environment, well, to the environment in general. And their agency for dealing with the radioactive sites, what he does to predict the transfer of radiocarbon from nuclear power plants to marine organisms. Well, why would we be interested in looking at radiocarbon anyway? It's a naturally occurring radionuclide. It's all around us. It's inside every one of you. It's also produced by human activities, nuclear weapon testing, civil nuclear power. It has a really long half-life, 5,730 years. And it behaves like stable carbon in the environment. So inorganic C14 will be uptaken during photosynthesis and then end up into the food chain. It doesn't bioaccumulate, so the same as the microplastics that Natalia was just talking about. As a result of its long half-life and how it behaves in the environment, it's estimated to be the largest contributor to the global collective dose from civil nuclear activities. What that means is that every single person globally has a tiny bit of C14 in them, which comes from nuclear power. It's not doing anything to you, but when you add up that tiny amount in everybody, it becomes a number that people that like to add up numbers go, wow, that's a big number. That is true. So we're working with the Kattegat Ecopath model, which contains about 30 functional groups and was developed to describe the ecosystem in 1982. And it has also been fitted time series in EcoSim to 2009 with a series of different forcing data. And we are looking at EcoSim and EcoTracer together and projecting forward to 2025. We have an ecospace model as well, a large list of different environmental drivers that are using to force distribution of our biomass in this area. And we do, and I will show you some results from EcoTracer and EcoSpace model, but this area in particular does require further calibration, further work, which Maciek, I think, is hoping to be able to do. We're also incorporating advection in our EcoSpace model in order to disperse our C14 when it enters the environment from point source at Bath Fjord. We're actually only using a single map layer for average advection in the model, which is different from some stuff that I did before, where we utilized the spatial temporal framework as well to change that through time. This captures an important northward current in the eastern Kattegat, where the discharges are made. But then the low resolution in this instance means that we don't actually see the fjords where discharges are made, or also all the neighboring fjords in this area. So this is an area where a high resolution map could be helpful. I won't go into too much detail on the parameterization of EcoTracer or the equations and stuff there. Natalia's already done a really good job of that. The only thing I'll mention is our inflow in here. So we have two sources. We have an atmospheric release of C14, and we also have a marine, direct to marine discharge as well. Generally, our parameterization that we're using for uptake C14 in here is based on work that I did previously in the Irish Sea. So there's lots here. You don't really need to see or know everything that's going on here. So this is some results from our EcoSim with EcoTracer model projected 2025. So time on the x-axis, on y, we're utilizing the unit activity and becquerels. And what we're seeing, so we did Monte Carlo routine, 400 different iterations, changing the starting conditions very slightly. We're incorporating marine discharges as well as atmospheric. Atmospheric component is significantly larger here. The output that we get is the above background activity as a result of Ringels discharges only. And this is in EcoSim, an estimate average activity for the entire Kattegat area. Across all our functional groups, we get a range of between 2 and 20 becquerels per kilogram of carbon, and a mean of 4 to 7. Just to put that into perspective, that's up to 3 orders of magnitude less than what's observed in the Irish Sea. And in the Irish Sea, we deem that to not be radiologically significant. So obviously that is also not radiologically significant. In ecospace, again, lots going on here, but you don't really need to know what's going on in all of these boxes. So exact same time and activity on our y-axis. The model projected forward to 2050, we looked at a couple different RCP projections. I'm not going to go into the details of that, but if anybody wants to ask why after, then please do. We're only considering marine discharges because we want to know what's going on in that local area proximate to where discharges are made. And so this is, it's called Region 1, is an area 64 kilometre squared region that's proximate to the discharge point in our ecospace map. Where there's no biomass, we get no activity here, which is nice. And we get some, so here's Sol in this box, Sandeel here, where we're seeing some relatively high numbers. So we're going up towards 200 becquerels in these boxes. What was interesting as well, our route to get C14 into these fish has to be uptake by primary producers down here at the bottom. And we don't really see any activity in primary producers. So that was a bit strange. Oh, yeah, I highlighted them. So we had that box proximate to the discharge point, region there. We actually had several regions across the map, but then one of the others is up at this other fjord, just to the northern portion of the north of where the discharge point was. And so if we start looking at actually what was going on in that area, and actually this is easier to look at. So just some of the functional groups that are of interest. And we start seeing these higher activities in our primary producing groups. And that was basically because of advection in the model, where we're seeing an accumulation up where that fjord is in space. But we didn't see the accumulation at the discharge point because our resolution in the model meant that we couldn't get an accumulation happening in that area. The advection was just carrying our discharge either way. So we start figuring out, okay, so what is happening is that we had movement of higher activity fish from one region to another, and that's why we're picking up those higher activities in another region, and they're picking it up in a different area where we can see these higher activities in primary producers. And so we're seeing a similar range up to about 200 becquerels. And then start wondering, well, does that warrant investigation then? And happily enough, the same funder had decided the same thing at the same time as they funded this project and started, I think the actual work started in 2020, 2021 to actually collect some samples close to the discharge point and do some analysis on what's going on. So this is ongoing work by Professor Christina Stenström at Lund University. This figure is different units, but I've kind of recalculated them in the text over on the right. And so what we have here is mussels, both the shell and the tissue, and fucus species seaweed, macroalgae. And the range that they are getting in seaweeds there is 24 to 240 becquerels above background, so similar to the range that we're getting in our Ecospace output, 0 to 200. Although they did then very recently go and find a fish, which was very, well, not very active, but relatively more active. Shouldn't say very, that scares people. But I have to keep in mind that that also includes potential other sources of C14 in that area. Despite being really close to a discharge point, there are other sources, other anthropogenic sources, whereas model, we're only considering ringles. And so the other thing to say is that in Ecospace, where we're getting these, we're at the lower end of the observed data for estimates for macroalgae and mollusks. But then there is obviously resolution limitations. These samples were taken very close to a discharge point. In Ecospace, we were looking at a region that was 64 kilometer squared. So, you'll be pleased to see that slide. So some general conclusions based on that work. So the discharges are predominantly to the atmosphere. When considering the entire Kattegat, atmospheric releases are the most significant to consider. And that was easiest to be able to model in EcoSim. The marine discharges become increasingly more important as you move closer to source. Makes sense. And obviously, if you don't know what the environmental concentrations actually are, then you need to start thinking about dispersion, advection, and potentially doing this in Ecospace. The range and observed activities was modeled, maybe not for the same biota or functional groups, but we did indicate that where an accumulation of C14 could occur in the environment, that the activity in the biota warrants further investigation. And obviously, resolution versus reality is a balance. At what scale do you need to make your predictions for? And then do you have enough data to be able to go to that resolution? And any questions? Questions? Or you could ask me over a beer.
Thank you. Thank you very much. Okay, so before we have that beer, I have two things. The first thing is that I have a few people for which you cannot have a beer because we don't have your slide for tomorrow morning's pitch presentation. I'm going to name and shame them. I'll start with Santiago and Igor. Do not let those people drink any beer because tomorrow morning it'll be a mess. And Gabriel Barros Gonzalez de Sosa. Gabriel, are you here? Maybe they're not here yet. And Jayshree, is she here?
She had to head out, she had to nip out.
Oh.
But she is going to be back.
Okay, but we need her slide for tomorrow morning. So
I've seen it, I know it's there.
but can
Make sure you
yeah. Before you have a beer, you are not allowed to have a beer either. Oh, excellent. And the second thing is that we have we don't have on the slides all the students. So Kim is going to come up and explain the process which we offer.
Well, really what I want to do is put your attention to this QR code. We did not assign judges to judge student presentations. So it's all of you. What we've done with the presentations is that you see the little hat when they're students. Not all the student presentations had hats. So if you are a student and you see that's not indicated, please say that you are a student and you will be incorporated. I think what we'll do is have a list of student presentations just all together on a slide tomorrow, and that will make the judging easy. You'll see these next to posters as well. The QR codes will be next to student posters. It's all the same QR code, though. You could scan that QR code next to a poster and judge a presentation. You're allowed to judge more than once because you're going to forget names and whatnot. Just indicate your own name as well, and then we can all figure out on the back end who got the most votes. There's a comment section, so if you vote for more than one student, see if you can rank them, and that will all help with us with the evaluations. One question I put in there is the first 3 words of the title. That can be hard to remember sometimes. Just put something in there. It's really to make sure you didn't like mismatch the presentation and the student. So put a keyword in there, look at the program. All the questions are mandatory, so put it in the little survey. There'll be 1 prize for a presentation, oral, and 1 prize for a poster. So we would like all of you to participate in this, even when you're a student yourself. And again, you can judge more than once. Just always add your name to the form when you do. I think that's it. We are allowed to drink beer, except for some of you.
Yeah. Yeah, well, hold on.